I've got there are a couple of audience questions here I want to touch upon before we move on. So it looks like, as we're talking about prophylactic vaccines, as we're thinking about even the cancer immunotherapies, there's a question here. mRNA companies have gone into phase 1 with a multivalent monotherapy approach involving 3 to 4 mRNAs. Any sense of how the agencies view how many different mRNAs will be too much in a multivalent monotherapy approach? Tiffany, maybe you want to tackle this one. I know the concept of how many mRNAs is too much is a topic of good discussion.
Right. So I'll start by saying, be very careful about how you compare your product and your clinical study to others, because, most of that information is going to be confidential. What you see in public may not be, a reality. So what really matters is that you understand your program and your product best. And there are basic requirements for entering phase 1 studies, and there are requirements for what's necessary to proceed with in the US, the BLA supporting studies. So what you're going to use to apply for licensure with. So if you're going to come in and say, I'm going to use between 2 to 20 targets here, different mRNAs, well, how is that going to help you inform your dose and your safety that you're working on in your phase 1 clinical study? Are you going to have like a maximum amount of mRNA that you're going to develop to administer to a patient? Are you going to divide that by 2? Or are you going to divide that by 20? And if a patient has a safety signal, how are you going to be able to look at your product and figure out where that signal is originating? Is it some aspect of the product that's resulting in, let's say a grade 3, grade 4 event in a patient? Also, how are you going to figure out what characteristics of those products are resulting in efficacy or a patient response, say, to a malignancy? So how are you designing that study to fit the requirements of phase 1 and phase 2, 3? You need to have that thought out and a well-justified plan to the agency.
Great. Okay. So making sure that, again, it comes back to your justification, right? And perhaps being specific, not just giving a broad range of potential targets here.
And patients deserve that, right? Patients deserve the chance for potential benefit by enrolling in these clinical studies.
Mm-hmm. Absolutely. Ulrike, do you have anything to add to this question?
Yeah, I mean, I doubt that it's that you can just throw in a number and say like more than 20 will not work. I think you need to have a plan and a kind of concept on what the range of antigens is within your control, and how you would delineate any kind of clinical responses that you could have to your product. So if, like Tiffany said, you would have a range from 2 to 20, I mean, that's a tenfold difference, right? Are you able to control this difference and be able to delineate any kind of response that you would see to your product?
Mm-hmm. So being able to know.
If so, how? Yeah. Right?
Mm-hmm. Sorry, I cut you off.
And this comes, this comes then also in whether you have the statistics in your clinical study to actually be able to see any potential differences if you have either 2 or 20 antigens involved there, right?